Okay, so we are now recording, and thank you very much for agreeing to give a Build a Cell seminar, and
Sure.
you can get started.
Yep. Okay. You can see I guess you can hear me for sure. Can you see my slide?
Yes, we can see your
Yeah.
slides.
Okay. So here's the title of my presentation. I try to make it fit the seminars, but it may not. So because I think a kid contacted me because of the cell-free expression system, and we use this as a presentation in the fungal genetics conference at early time of this year. But actually, today's presentation is not only limited to the cell-free systems and the use of the cell-free systems. So I guess I have some other tools, and I have more stories about the fungal arsenals used for wood decomposition. So, and talk about me. So my name is Jiwei Zhang, and I'm assistant professor at Department of Bioproducts Biosystems Engineering. So my department is at the St. Paul campus. So this is my experiences. I got my PhD in microbiology in China in Shandong University. And I joined a lab here to continue my postdoc training. And so my advisor name is Jonathan Schilling. And if you know him, he's a person to study fungal ecology, and who is, the main interest is to study the wood decay fungi and how the wood decay fungi impact the ecologies. I take the postdoc training by using some system biology approaches to understand the molecular mechanisms of wood decay fungi, how wood decay fungi decay, depolymerize the lignin cellulose structures in the plant biomass. So, and then I take the assistant professor job at another department, my current department. So, and I started it about 3 years ago, I think, so right before the pandemic. So I try to, and I'm still trying to develop my independent research. So I'm thinking of, I can talk about my research today. You can have some understandings about my independent research. But instead of, studying the ecology, so my current research is more like to study the molecular biology or genetics of the wood decay fungi. So my research is mainly focusing on the wood decay fungi. So because they are important to the ecosystem, and they are the main drivers of the carbon cycling in the ecosystem, because these fungi can efficiently decompose and metabolize the carbons sequestered in the plant biomass. And they can recycle the carbons into the atmosphere as CO2. So they are very important to the sustainable ecosystem. And they have many applications. So for example, at the beginning, Kat and me are talking about Mars colonization. Think about it for example, one day if we migrate to the Mars, we probably wanted to build our ecosystem on Mars, on another planet. At that case, probably we will need to build our own sustainable carbon cycling system. And then we will have to probably include the decomposers and decomposer fungi to achieve the carbon cycling process on another planet. So that's one of the applications, but another application is very clear, is we wanted to convert to the plant biomass, which are sustainable carbon resources on our planet. We want to convert these sustainable resources to sustainable biofuels and bioproducts. And fungi are important in this process because they can degrade the recalcitrant structures of the plant biomass, and they can convert the carbon sources to other format of carbon sources in the biofuels and bioproducts. So let's see the fungi, especially the wood decay fungi, they are very important to the circular economy. So sometimes I see fungi, and sometimes I see fungi. I think it's basically the same thing. Most of the time I call it fungi because it's interesting. It's a fungi. Anyway, so here are some of the examples of the wood decay fungi in the forest. And they are very commonly seen decaying wood in the forest. But whenever you see the wood, and you can see the wood decay fungi, it's definitely because one of the common sense we believe is wood are never stand, they will be rotted. So the wood decay fungi are the key factors that causing the wood rot in nature. So this is my lab goal. Actually, we want to use a fungi to build a clean and sustainable world. It's very ambitious, but I just want to make it a little bit interesting, and which will help my management and my team leading in the lab, right? So, and basically we're using some system biology approaches and gene editing tools to dissect the fungal capacities used for involved in the wood decomposition or plant biomass decomposition. And so we wanted to convert these plant biomass polymers to make a sustainable bioproduct. And on the other side, because many pollutant polymers are having the very similar structures to the lignin cellulose plant biomass. So basically, many pollutant polymers are carbon-carbon bonds polymers or other structures that are resembling the structures of the plant biomass. So studying the wood decomposition process will also help us to degrade the pollutant polymers that are generated by the human beings for environmental remediation. So our goal actually is, can be described simple as understand the mechanisms and then harness these mechanisms for the relevant uses. So this is a very detailed top-down design of my lab. Is trying to make a fungi a more colorful use. So I use different colors to color code the different research in my lab. So there are basically 1, 2, 3, 4, 5 main research in my lab. So, and all the researches are for sure based on the wood rot fungi. So first, we wanted to develop genetic tools to understand the fungal mechanisms. And so we're using some reporter genes to develop the genetic tools in our fungi. And as a little bit background, no research or no groups in this world have really developed the genetic transformation or genetic manipulations in the our model wood decay fungus, which is a brown rot fungus. I will talk about the brown rot fungus later in today's presentation. The brown rot fungus or fungi are a very unique group of fungi. They can very efficiently decay the wood, so that's why we're interested in this group of fungi. So after developing the genetic toolkits, we want to understand the fundamental genetic mechanisms used by wood decay fungi to decay wood, right? And basically we wanted to decode the genetics of this wood decay fungi. And this is using the pure cultures and pure wood decay fungi. We're also interested in the socials of the fungi. So, and one of my project is trying to understand how my wood decay fungi interact with other wood decay fungi and bacteria during the wood decomposition process. So as one of the applications, the third areas in my lab is trying to use a fungi to clean the pollutants as a micro-remediation method. So I use blue color and this brown shape and trying to code the application of the micro-remediation. And the last piece, which is probably my future area. So we wanted to use our fungi as a platform to develop the synthetic biologies. So there are several reasons to do this. One is this fungi, they can convert the plant biomass as sustainable carbon sources. So that means they can use the plant biomass as a carbon source, and they can convert this carbon sources to any other bioproducts and by the synthetic biology designings. And also these fungi are higher fungi. So they have some unique metabolic pathways that are not used by other fungi or other organisms. So they have their unique needs to convert and produce the unique bioproducts. For example, some of the secondary metabolites are uniquely synthesized or produced by this higher fungi, the wood decay fungi, so which cannot be produced by other organisms. So this will give us a great opportunity to use wood decay fungi as a platform to convert the cheap and easy to obtain carbon source, which is plant biomass, to other value-added bioproducts. For example, the secondary metabolites or any other valuable product. So this is the last piece I'm really interested in my lab in the near future, is trying to develop the wood decay fungi as a platform for synthetic biology. Usually this is how we understand the functions of wood decay fungi, which is I call it multi-scale functions, from the whole planet ecosystem to the forest ecosystem, and to the interactions between fungi and to the single species, and to the cellular level and gene level. This is usually how we understand it. And my lab is not a such a big scale levels. And actually, this is my previous lab I was doing, trying to understand the functions of the wood decay fungi to the whole ecosystem, to the carbon cycling process. So here's my lab. So we wanted to decode the fungal phenotypes at a species and cell and gene levels at a relatively smaller scale. So in nature, fungi evolved in many ways to degrade plant biomass. And you can see clearly while you are hand out to the forest, and you can see a lot of fungi, actually, they're decaying the wood and decaying the organic matters. So, for example, this include some mycorrhizal fungi. They're growing under the ground. Actually, most of the fungi parts are growing underground, right? They are in the format of mycelium, so they are underground, and you cannot see the parts you commonly see are the mushrooms or fruiting bodies, right? But actually, the underground parts are the big parts that are contributing to the nutrient cycling process. So mycorrhizal fungi are one of the fungi, so they can grow together with the plant, And so they can form some symbiotic relationships. And these fungi actually, they have a limited capacity to decompose the plant biomass because they have to form the symbiotic relationships with the trees by first colonizing the trees and roots. So they have a limited abilities to decompose the plant biomass. And pathogen fungi, clearly they also needed to decompose the plant biomass before they can infect the trees, right? So they also have the plant biomass degrading capacities. Also, there are at least two groups of fungi or wood decay fungi, we call it. They are very efficient plant biomass decomposers. So they are white rotters and brown rotters. So these two are main plant biomass or lignin cellulose degrading fungi, probably in the forest system. And they are the main contributors that can degrade the wood and recycle the carbons into the atmosphere. So viral fungi actually, these two fungi, they use different ways to decay the wood. And viral fungi actually, they can decompose everything. They can degrade everything in the wood. And if you know the wood is composed of the lignin cellulose structures, and lignin cellulose is composed of cellulose and hemicellulose and the lignins. So these wire rotters are very efficient. So they can degrade everything, so hemicellulose, cellulose, and lignins, because these are all carbon format polymers. So on the other hand, the brown rotters, they just selective, they seem more smarter. So they can selectively degrade the carbohydrates, which is the cellulose and hemicellulose, the main carbohydrate. But they don't like to eat the lignins, because the lignins are having a most recalcitrant structures and very, very difficult to degrade. So that's saying brown rotters might be relatively smarter comparing to white rotter, at least in my sense, right? So, but actually we cannot judge fungi, according to our opinion. But these fungi, these brown fungi, so they can very efficiently decay the wood. So seeing if your residential building is infected by the brown rot fungi, you gotta have a big problem, because they can quickly damage the structures of the wood. And they're having a very efficient way to decay the wood structures. So if you go to the forest area, especially here, so you will find many brown rot wood residues here in the forest, on the forest floor. And so they are basically in the brown color. That is because of the lignins that are not eaten by the brown rotters. And if you take one piece of these particles, and if you squeeze it, you will feel the power of the fungi, because you can easily fragile these particles into a very minor powders. So that's the beauty of and the power of the brown rot. So that's why we're interested in study brown rotters, actually. So here's one of the images I took from Cedar Creek. And this is a brown rot, and you can see a clear cubical structures and cubic structures of the brown rot. If you take one piece from this cubic and you squeeze it, you will feel the power of the brown rot. So, and that's my words, brown rot are probably the most efficient wood composer fungi. So I call it most efficient is mainly in terms of their abilities to fast decompose the wood structures. So actually, you can give different standards to compare the brown rot fungi and white rot fungi. But at least, to my end, I thought the brown rot fungi are most efficient wood decomposers because they can, most efficiently decompose the wood structures. And this is the extensively decayed wood by the brown rot fungi. You can see here is almost a powder and like a soil. And this is electron microscope imaging trying to show the structures, the damaged structures of the wood cells by the brown rot. As you can clearly see, it's kind of like, completely deconstruct the wood cells structures, wood cell walls structures. These are two commonly used brown rot species in our lab. So the left one is a polyporous, and because it has many pores, it's called polypores. And the shape is very like a placenta. So, and they name it as a Rhodonia placenta. This is one of the model fungus used in our lab to study brown rot mechanisms. And also this is the first brown rot species that are got sequenced. So that, if I remember correctly, the genome of Rhodonia placenta was obtained at 2009. So almost 13 years ago, 10 years ago, right? Many years ago. So that's good because we can have the genome, we can have many studies to decode the genome, the genomic information. So this is another brown rot fungus we commonly use in our lab, which is a model fungus. Actually, we can genetically, right now we developed a system or platform that we can genetically manipulate this fungus. This fungus is a Glyophyllum trivium, and which is also commonly seen in Minnesota area, in Midwest and northern part of the world. So this is how powerful the brown rot is. So I compared the decay rate by measuring the mass loss of the wood blocks used for decaying by brown rot and white rotters. So clearly you can see the brown rotters are causing a fast decay rate and mass loss of the wood than the white rotters. And another interesting thing is if you measure the free sugars that are released by the fungi during the decay process, you can see clearly here the brown rotters they can generate more free sugars than the white rotters. So this is a sort of indicator that the brown rotters are more efficient in terms of extracting the sugars of carbohydrate from the wood. But you may ask a different question, later in today's presentation, why these fungi can release many sugars to environment. They are not so selfish, right? And that's gonna be not economic because think about it in the real environment. If you release the sugars to the environment, these sugars can be easily uptook by other microbial species, right? It's gonna be a non-economic thing to do. But this is, but I guess, probably this smart fungi, they have evolved other strategies to protect their sugars, their food. But this is the brown rot mechanisms that are being understood since 1960s. So, and people trying to track the brown rot process, we got some understandings about the brown rot phenotypes. So during the decay, as brown rot fungi attacks the wood, they will first cause the mechanical strength losses of the wood. So this is what I'm seeing in the brown rot. If you find a brown rot problem at your home, it's gonna be a big problem because this fungi can quickly cause the mechanic strength loss, means they can quickly depolymerize your wood building structures and without eating the carbohydrate. So it's after the mechanical strength loss step, so this fungi will start eating the carbohydrate in following the order of arabinose and galactans and xylins, which is mannese, which are hemicellulose. And finally, they will eat almost all of the glucans, which is made of glucose. And later, after that, they will leave the modified lignins, which is a most recalcitrant part of the wood. And so that's also why you've seen brown rot brown rotted wood are in the brown color. That's mainly because of the colors from the lignin residues. And these lignins, of course, are modified lignins, which is different than the original format of the lignins in the wood. So, and evolutionarily, so this brown rot fungi, they evolved multiple times from the different main clade of the fungi of the Agricomycotina group, and that's a convergent evolution. That means, you know, they are evolved from different orders, and so their siblings are in each order. You can find a lot of wire orders. So that's why we see in the convergent evolution. So if you think about the distance of the number 1 brown rot group and the number 3 brown rot group, actually they are from the different orders, right? If you think about you know, the distance, this distance in terms of human beings and other animals, this distance is more like the distance between human beings and a rabbit, right? So hopefully that can give you some clear views of you know, about this relationships of this convergently evolved brown rot clade. So this brown rot evolution actually is shared by some of the common pathways. And so this brown rot fungi during evolution, they lost many lignin cellulose degrading genes, which are commonly or traditionally used for decaying the lignin cellulose. And for example, they lost many cellulosis genes, which is used for degrading celluloses. And they lost the ligninolytic peroxidase genes, which is used for degrading lignins. So that's very weird, you know. Although they lost these genes, but they still can efficiently degrade wood structures. So that's kind of like a mystery in the field we're trying to understand. So according to the molecular clock, actually, we can see that these brown rotters, they are evolved about 150 million years ago, which is about a Jurassic time. And so if you think about it, here's something interesting, you know, you may interested to the brown, may raise your interest to brown rot. So 150 million years ago is, you know, is like this map. Our Earth looks like a 2 main continents. This is today's continents, today's Earth. So the brown rot fungi actually they evolved when our Earth or continents are organized as two main continents. So and the definitely the environment at that time, like climate, temperatures, oxygen levels will influence the brown rot evolution. But we still don't know how that influences today's distribution of the brown rot. It's very interesting because usually you only find the brown rot fungi in the northern part. I think it's the north of 35 degree, but not in the tropical areas. In the tropical areas, you find almost all fungi or wood decay fungi are white rotters. So that's kind of like another mystery. We don't understand, why the brown rotters are so efficient, but their distributions are limited in nowadays. So only at that northern part. So it's maybe this is, some researchers and said maybe this is related to the tree species. So because in the northern part, there's only coniferous trees, right? So they suspect that the brown rotters are related to or associated with the tree species, but this is not validated yet. So this is a mystery, so why brown rot are so efficient or super efficient at decaying wood structures, but their distribution are limited. So and also if you think about the Jurassic time, which is a dinosaur's time, right? So and another interesting thing I want to talk about is when we're looking at the movies and the images, pictures, this is what we're seeing. And dinosaurs and coniferous trees and very high trees, but we never see the mushrooms, right? And so I think that's probably a mistake. And probably, in the future, we want to add the mushrooms and together with the dinosaurs and raptors here, and so that's probably a better description of the ecosystems back to the Jurassic time. So and probably the dinosaurs, they are using the mushrooms as a food, right? So when I was watching the Jurassic 4 weeks ago with my son, they're talking about trying to recreate the dinosaurs and build the dinosaur parks in our world. But I don't know if they're also thinking about trying to bring the mushrooms back, and because that is probably is a part of the ecosystem of the system where the dinosaurs are living. But anyway, so that's back to our main question today. So that's something I was wondering some of the questions and things I think maybe is interesting to you today. So that back to my main question today. So why brown rot are so efficient? So it's because they are using some bones, and which is given by the reactive oxygen species. So these reactive oxygen species can be used for the electrophilic attacks, which can unspecifically and aggressively attack the lignin cellulose structures. And these species can include, for example, the hydroxide radicals and which is a very strong radicals that can aggressively attack the polymers. Also, some other radicals that are derived from the hydroxyl radicals. And this has been proved by many research in the previous days for the brown rot fungi to use reactive oxygen species ROS to decay the wood. And this is proposed since 1960s and 1970s. This is an equation used for producing one of the ROS radicals is the hydroxyl radicals, which is mediated by the reaction between hydroxide peroxide and reduced iron to produce these radicals. These radicals can aggressively attack living cellulose structures and cause the degradation. And this ROS actually can be generated by electron relays. So basically the cells, this is a middle part. This is the wood cell wall, the yellow part, and this is a wood cell lumen. And in the wood cell lumen, the fungal cells will grow in and penetrate the wood. And they will start secreting the metabolites to decay the wood, metabolites in enzymes. The ROS actually is generated by the electron transfer process or electron relay process driven by the fungal activity. So, and these fungi actually, they can synthesize and secrete some reductant, which is carrying the electrons. So this reductants also can chelate the irons and then reduce the irons to iron 3 to iron 2. So these electrons thus are further transferred to the reduced irons. So also this metabolites, some of the studies have shown that this metabolites are likely hydroquinones or some other low molecular phenolics. So these metabolites also can send the electrons to oxygen to produce hydrogen peroxide. So thus they have other reactants for the Fenton reaction. And then by the Fenton reaction, the electrons are further transferred to hydroxyl radicals. Hydroxyl radicals will cause the electrophilic attacks on the wood polymers. So basically, this is fungal activity-driven electron transfers, and that can cause the production of the ROS radicals and eventually degrade the polymers. So, but what are the problems of using this ROS? I don't know anyone are still there. I just, pause here for a second, ask this question. What are the problems of using reactive oxygen species to degrade a wood?
I don't think you have enough chemists on the call.
Okay. Right. I would just, So this ROS, or because they aggressively damage all the polymers, right? These polymers would also include the cell structures and enzymes. So that means these ROS can cause a self harm. So they can damage the cells and fungal enzymes, right? So although this is a very efficient, or although they are making sense to use this ROS to decay the wood, so there is some conflict still. So that means fungi should have evolved strategies to avoid the self-damage caused by the ROS. So basically, there are several possibilities or explanations of these strategies. And first, so you would think you wanted to separate these ROS productions in a distance from the fungal cells and fungal enzymes. Basically, there are two hypotheses. One is you can change the macro environment surrounding the cell. And for example, you can change the ions concentrations, the pH, and that way you can control the ROS production only at the substrate, only on the substrate, but not close to the fungal cells. And the second thing you can do, the fungi can do, is to temporarily segregate the ROS productions. So they can, produce, for example, the ROS is at the early time of the decay, while there is no plenty of fungal cells are growing. So there are also some other strategies fungi may use to avoid the self-damage of ROS. Is trying to evolve their enzymes to produce some better enzymes or more enzymes. Seeing if better enzymes basically means you have the enzymes that can resistant to the ROS damage. More enzymes means if you have a more and you damage some enzymes, you still get quite a lot of leftover enzymes that can work on the ligand cellulose degradation. The third possible strategy might be fungi can use some antioxidant. So that is also possible, but that's not involved in today's presentation. We did give some studies on this possible strategy. So first is macro environment control. So that means these metabolites, once synthesized, they needed to be diffused to the wood cell wall. So this diffusion probably will cause some gradient. This gradient probably can only restrict the production of the radicals immediately on the substrate. So, and another big factor that can be used for this macro environment control is a pH, because Fenton reactions will only happening at a certain pH. So it has a optimum pH at 4. So that means the macro environmental factors may control the productions of the ROS at a limited place or at a restricted place. But the problem is it's really, really hard to test this hypothesis because the metabolites can diffuse very quickly in this environment between the cell and the wood substrate, and between the fungal cell and the wood substrate. And also the concentration would be very low. It's, I think it's beyond my expertise. I don't know what kind of probes we can develop to measure, for example, the iron concentrations, the metabolites concentration, and pHs. So that's saying, this hypothesis is very hard to test. So instead of testing this hypothesis, I tried to test another hypothesis, is a special temporal segregation of the ROS production. To do this, actually, we let the fungus to decay a piece of wood, and we let it grow along the wood wafers. We call it wafers, or wood wafers. And then we can separate the early decay stages from the late decay stages. And we can track the activities of the fungi or wood decay activities along these different decay stages. And we use RA-seq technologies to understand the genes expression in the fungi. So we found actually that the ROS producing genes are mainly expressed or mainly upregulated at the early decay stages, and while the glycoside hydrolyses are mainly upregulated at later decay stages. So these glycoside hydrolyses are mainly involved in the polysaccharide or cellulose or hemicellulose degradation, which is used for degrading these polymers for releasing the carbohydrate. So, and this is a very, then we proposed a model, we try to explain this ROS production as a two-step mechanism. So the first step is the fungi will turn on the genes expression related to the ROS production and then turn it off. And then after the ROS depolymerize the overall structures, and the fungi will turn on the expressions of glucoside hydrolysis and then eat the sugars mainly. So if you look at this phenotype or progress, decay progress diagram, it's making sense. The ROS at the first cause the mechanical strength losses, and then the polysaccharide hydrolyses are produced to extract the sugars. So we call it two-step. So, and later on, we tried a different species, and we found actually a very interesting thing is this two-step is conserved in some of the brown rot species, but not for all brown rot species. That means probably, these brown rot species are from a different clade or evolved different mechanisms for the ROS control. So there are several possible explanations actually. So the first is probably the GH enzymes, the hydrolysis enzymes in some of the species, they are evolved to be tolerant to the ROS. And the second thing is clear, probably the predictive function of these genes and genomes are not equal to the real functions. Also, it's also possible there are some other ROS producing genes we're not knowing yet. So to study these possibilities, actually, we tested the second possibility, hypothesis for avoiding the ROS damage. So we actually, this part to use the cell-free expression systems. So we choose some of the enzymes that are fitted into our ROS producing model, and they are early upregulated enzymes. And this includes some ROS generating enzymes and some other glycoside hydrolyses that are produced at the same time of ROS. So we use a cell-free. I guess this part is the part that cats call my name in the fungal genetic conferences materials. But we want to characterize these enzymes, these brown rot enzymes functions in a faster way. And back to that time, we're thinking of using yeast expression system to express these enzymes and characterize them. We're also thinking of using E. coli. But the thing is, brown rot are higher fungi. They might have a very different expression system than E. coli, which is a bacteria. Yeast, we tried yeast, but we tried 17 or 15 genes, enzymes, but we never get a success. That's very interesting. And then we switched to use this cell-free enzyme expression system. And we think it's just faster. And actually, this is a collaboration between my lab and the PNL, Pacific Northwest National Lab. Irina and James are two main people who are helping us on this project. So we express these enzymes, and we still have, a very low success rate using this cell-free expression. Actually, only 25% of the enzymes can be successfully expressed to have the activities. This includes some benzoquinol reductase. If you still remember, this is the enzymes that are used for producing hydroquinones. Hydroquinol is the fungal metabolite to carry the electrons. And a ferric reductase, which is enzyme to reduce the iron 3 to iron 2. And also some of the other hydrolyses are also successfully expressed to have the activity. So we characterize the enzymes activities, and then we characterize the pH and other characters of the enzymes. And basically what you're seeing here is this is one of the hydrolyses is called alpha-L-arabinophrenosidase. It has a very robust capacities to resist many conditions. The pH, they have a very wide range of pH stabilities and temperature stabilities comparing to the other three enzymes. And also this alpha-arabinophrenosidase, this hydrolysis, actually it can it's not performed good under inhibitor conditions, but most importantly, this hydrolysis can resist to the OS damage. The white bars are the controls, and the black bars are the enzymes activities that are subjected to OS damage. So you can clearly see this hydrolysis can be tolerant to the OS damage. So that means brown rot hydrolysis, they involved novel activities to resist, to tolerate to the OS. So that's the thing, by using the cell-free system, we characterize 4 enzymes that are important to the brown rot process. And we'll also find their characters. And importantly, we find the evolution of the hydrolysis. And so this evolution is, evolved towards making more making enzymes more OS tolerance. So, and we also tested another possibilities as the brown rot can invest more energies to produce more hydrolysis, and this is about RAC. So, but however, the predicted functions is not equal to the real functions, and in vitro functions by the cell-free system is not equal to in vivo. So we decided to do the more genetic studies to link the brown rot phenotypes to genotypes. So thus we developed the brown rot transformation system, and we heterologously expressed a lacase gene in our brown rot system, and then we developed this lacase system as a reporting system to further optimize the G-Trivium, the brown rot platform. Currently, we're doing the CRISPR-Cas mediated to the editings. So we have expressed the Cas9, and thanks to the cell-free system, we will more specifically target the enzymes that are identified by the cell-free system. So this is a story about the brown rots. So I just write it down, I think, here. So it's about 150 million years ago when the Earth only had 2 continents. A group of fungi evolved quietly and slowly. So they extremely like to eat the sugars of the wood. To do this, they abandoned most of the conventional weapons developed by their ancestors Instead, they started to use a more powerful but dangerous ROS weapon. Then the weapon evolution happened. So they are added a smart controlling system to precisely send the ROS to a target to the wood substrate without self-hurting. So partnering with this new weapon system, a diverse of old enzymes are banned and shuffled to resist to the ROS self-harm. So, and all right, and with this, using this updated weapon system, this fungi started their colonization on the Earth. So I think this is a story I want to end, today's presentation with this short story. But the story is continuing, right? It's not ended because we're using the different approaches to further understand the brown rot mechanisms. These are the lab members and collaborators and founders that are involved in this research. So, and also we're hiring the students who work on more brown rot genetic studies. So that's it. Thank you. Great. I think yeah. Yeah.
Thank you so much.
Yeah.
There is a lot of questions in chat. Can you see chat?
Yeah. So
if you could read a question before answering it, read it out loud. That's going to be for people that watch the recording.
Yeah. Do we have time? Actually, I think we're at 12. Sorry about that.
I think a lot of people will drop off if they have to, but I think some people stay to listen. Okay,
cool. So we can answer questions one by one. Cell-free stuff always fit. I'm excited for the talk. Thank you, Dan. And Tanner, could the fungi rot be used to process the biomass for further wood processing, like making wood pulping naturally? Yes. Then the white rotters actually remember what I mentioned. So they can degrade the lignins. So actually the pulping process is a process to remove the lignins, right? So white rotters are actually, proposed to be used for the wood pulping process. And the brain question, did fungi change much since Cretaceous? I think so, because the mutation rate of the DNA, according to one of the Nature paper in fungi, the DNA mutation rate of the fungi is about a 10 to 6 one per 10 to 6 DNA base pairs will have a mutation from after one generation. So definitely the fungi will mutate. So, and change, yes, it will definitely change. And then other questions, you get a material degradation. I cannot quite understand this question. If Dan, you can explain this, what is the question? What is the meaning if you get a materials degradation?
I think, so I remember when that question showed up, I think this is the answer to your question, what happens when ROSs are produced? I think you had that question that you asked in the middle of the talk, what's wrong with making reactive oxygen?
What's the problem of using ROS?
I think Dan is answering that question.
Okay, yeah, okay, great. Thank you, Dan. Marcel, hopefully I pronounced the name correctly. Would the fungi cell be a candidate to build systematically, or is it very complex? It is very complex. So it's a eukaryotic cell. It has many subcellular structures, but fungi, it has been used as a filament, as a cell industry for many uses. It is used for enzyme production, secondary metabolite productions, but most of the cell industries of using fungi are using, for example, aspergillus and trichodermas. So these are well-studied fungal species. So these are model species that are used for as industrial cells. And Michael, could the ROS production be used to sterilize the materials? The fungi could eventually die out, but the processed biomass would be sterile, kind of like alcohol. I think so. I think so. And probably that's why the brown rot are having a success in the ecosystem, because the ROS is so aggressive. It's kind of like a sterilization to white up other microbials. Awesome talk. Thank you, Brandon said. You talked about a color tagging your project. That's cool. Do you express a different chromogenic or fluorescent proteins in? Yes, we expressed the GFP. So we already, we linked it together with the Cas9, we expressed it, but that's a codon optimized GFP protein. Anna said, can fungi that process would be made to extreme phylix, for example, to process biomass and low potential pressure radiation, and for that's a good idea. I think I think we can try to use genetic engineering to combine all of these good features into one fungus to make a super fungus. But actually, some of the wood decay fungi, they are extreme phyllic fungus. They can decay the wood in the very high moisture conditions, and also they can decay the wood in the Arctic areas, and some of the extreme conditions of Egypt, the desert. So they definitely they can, but that's a different species, right? So and so I think that's something we need to study to understand this extreme phylix of fungi, wood decay fungi. So I know Robert Blanchett, one of the old professors in plant pathology, he's studied this extreme phyllic wood decay fungi under the cold conditions or the desert conditions. So definitely, yep. I think that's all of the questions. Yep. Sorry, take a longer time.
No, I think everyone, I speak for everyone to say we really enjoyed it. I really enjoyed it. I learned a lot, and I mostly knew the sulfite work. I didn't know there's so much to care about brown rot.
Oh yeah. Yeah, if you walk into the forest, you see a lot of brown rot on the floor of the forest. So, and maybe you can identify them. You definitely can identify them very easily. So because they are brown color, yeah. Mm-hmm.
Thank you so much. Thank you so much for the talk again, and thanks everyone. And see you next week.
Yep. Thank you.
Bye.
Bye.